Hello everyone. In this section, we will go through some basic concepts in bioinformatics. The first concept is DNA. DNA is a helical molecule. It is a sequence of 4 nucleotides, or bases, that is A, C, G, and T. A is always paired with T, and G always paired with C. In human, DNA is a double helix, or a double-stranded molecule. RNA is typically a single-stranded molecule. There's one difference on the base between DNA and RNA. In DNA, the 4 bases are A, C, G, T. In RNA, they are A, C, G, U. There are multiple types of RNAs. For instance, messenger RNA or mRNA, transfer RNA or tRNA, ribosomal RNA or rRNA. What is a gene? A gene is sort of like a computer program. It provides instructions to the system in terms of how to build cell components or how to finish certain tasks. Each gene is a chunk of DNA sequence. One type of gene is protein coding gene. It provides instructions about how to translate the DNA sequence into a protein. There are actually other types of genes, but we'll only discuss protein coding gene in this class. A gene has many components, such as a promoter region, transcription factor binding sites, translation start site, exons, introns, and untranslated region. Here we have a picture for a typical human gene structure. This is a segment of the DNA sequence. To express the gene, a transcription factor needs to bind to the DNA sequence so that it can make a copy of the genetic region. Therefore, the first portion of this gene is the promoter region with a number of transcription factor binding sites. A transcription factor can recognize these sites and bind to them. Once the transcription factor binds to the sequence, it makes a copy of the sequence. What we obtain is a primary transcript. It's a copy of this sequence. The next step is called splicing, which removes those introns. Intron 1, intron 2 here are removed, and those exons are merged together. So exon 1, exon 2, and exon 3 are linked. What we have in this step is a mature messenger RNA. The part before the start codon is untranslated region, or UTR. Starting from this translation start site, this mRNA is translated into a protein. This is the translation. This protein can work with other proteins to finish certain tasks. What we just described in the previous page is the central dogma of molecular biology. That is, DNA is transcribed into RNA, and RNA is translated into a protein. A chromosome is a long DNA sequence. In the human genome, there are 23 pairs of chromosomes. They are labeled by its length. The longest one is chromosome 1. It has roughly 250 million bases. It is very long. That is why it is not easy to analyze those genomes. If there are only a few hundred or few thousand bases in each chromosome, it would be easy to perform the analysis. Chromosomes can be either linear or circular. Bacterial chromosomes are circular. Human chromosomes are linear. Genes are small segments in chromosomes. This picture shows human chromosome. You can see under a microscope. This picture puts everything together. In each cell, there is a complete set of chromosomes, which is the genome. In the human genome, there are 23 pairs of chromosomes. The total length of the human genome is 3 billion bases. If you want to store it, you need 3 gigabytes. In these chromosomes, some DNA segments are genes. For instance, in chromosome 1, there are roughly 2,000 genes, and the length of each gene is typically a few thousand bases. In other words, genes are just a small portion of a chromosome. These genes can be translated into proteins, and those proteins can interact with each other to finish certain tasks. Some of them can form a protein complex and interact with many other components in the gene. If you can clearly explain this picture, you have a clear idea about the basic concepts in bioinformatics.